So this is organic chemistry topic. Textbook reference is chapter 5.11. It's about amino acids and proteins. So we're going to start with amino acids. We're going to work our way through into proteins and enzymes. Right, so first of all, the amino acids that we have got to concern ourselves with, we have to understand, are called the alpha amino acids. They're the 20 amino acids which occur in nature. Okay, now there's only a couple of them you actually have to know. You do need to know what the definition of an amino acid is. So it's a bifunctional compound. You should have heard that term before. It's bifunctional. It contains two functional groups within the same compound. The name is a bit of a giveaway. Amino and acids tells us that we have an NH2 group. We've already studied the chemistry of amine, so this is also relevant to this topic. And the other functional group is the carboxylic acid group. And again, the chemistry of the carboxylic acids in A2 module 1 is also relevant here. Right, the key thing about an alpha amino acid is that these two functional groups have to be bonded to the same carbon atom. Now there are beta amino acids. They're not naturally occurring. They don't make up proteins, but they are separated. The two functional groups are separated by one carbon. No, so they're not bonded to the same. Okay. General structure of all amino acids are as so. So there we go. This carbon here, you may have picked up already, is our alpha carbon, and it's the R group that changes between one amino acid and another to give it its different properties. Two amino acids that you have to know off the top of your head. Okay, you won't be given these in an exam, are glycine, which is called amino ethanoic acid, where the R group here is a hydrogen, of course, and 2-amino-propanoic acid, more commonly known as its amino acid name of alanine, where the R group is a CH3. So they're the two simplest amino acids. You have to know both of them. You have to be able to draw them, write the structure. Okay. Right, there's just a little sample there of all the 20 alpha amino acids that do occur in nature. The green circle or the green section is the R group. So you can see every single one of them have all got an alpha carbon on them, but every single one have got a different R group. And that's what gives them the huge variety of different properties in terms of different proteins and enzymes, because when these all combine in the different variations that they can combine in, you get a monumental number of different variations. Right, so the two you need to know, as I've already said, are glycine and alanine, where you have the R group as an H and a CH3. So please make sure that you learn them. You will be asked maybe to draw alanine in its optical isomers, like so here, where the R would be, of course, a CH3. You may have worked out, if you go back to the previous one here, that glycine is the only amino acid that is not optically active. It's the only amino acid that around this carbon here doesn't have 4 different groups. So obviously you won't be asked to draw glycine as it's stereoisomers. So they are all indeed optically active, as you just said. They've all got 4 different groups around that alpha carbon, and they make non-superimposable mirror images, as you've done already in A2 module 1. So these should be pretty clear about that, and you know exactly what racemic mixtures are and rotation of plane polarized light and all of that. Okay. Right, so have a go at that question 511. Okay, just talks about what I've been talking about. Okay, and check your answers. Okay, again, have a go at that question. Check the answer. Right, so the acid-base properties, these, as you would imagine, they have acidic properties, they have basic properties, because they've got both acid groups and alkali groups, and they're said to be amphoteric. That's the term for something which can behave as both an acid and as an alkali. You'll find them in the blood. They act as natural buffers. They can remove acidity, they can remove alkalinity, hopefully for fairly obvious reasons. Okay, and they can maintain that pH. So what I'd like you to do is have a look at the one on the board, the generic one on the board, and see if you can write the structure of this when it is in an acid solution, and then write the structure of it when it is in a basic solution, when it is in an alkaline solution. So pause the video, have a go at that, and then come back and check the answer. Okay, so there you go. As you would expect, In acidic solution, the NH2 becomes protonated to an NH3 positive, an ammonium group, and in alkaline solution, the carboxylic acid group becomes deprotonated to become a carboxylate, a COO negative group. Okay, and that's the same for any amino acid. This type of question comes up frequently. Okay, so there you go. Have a go at that question for alanine. Pause the video, I'll move on. Same again, have a go at this one. Pause the video. Okay, again, have a go at that one. Right, so at a particular pH called the isoelectric point or the isoelectric pH, these compounds behave as what we call zwitterions. Now, also in the solid form of the amino acid, they behave as what we call zwitterions. Now, commonly we call these zwitterions. They can be called dipolar ions or indeed amphions. Three terms which are generally interchangeable, but zwitterions is the one that you will come across most with regards to amino acids. So let's have a look at what these zwitterions are, what they are, and what properties they confer onto our amino acids. So it is caused when the proton has internally transferred from a carboxylic acid group in it to the amine group. And if we have a look at the animation here, you can see what's happening. So I'll just run that again so you can see. And that's probably not that unexpected, because if you have a carboxylic acid and an amine group within a molecule, why would they not internally transfer? So you can see now we've changed the properties of these very much. We now have gone from having things which have intermolecular forces of hydrogen bonds maximum to ionic interactions and ionic bonds. And that's what a zwitterion is referred to as, this structure of an amino acid where you have a negative carboxylate ion and a positive ammonium ion. And this occurs only at particular pHs. That pH is different for every single amino acid. It's called the isoelectric pH, and it is also the form that amino acids are when they are solids. Okay. So zwitterions, dipolar ions, you can see here we just have the normal amino acid structure, and we're just trying to show the zwitterion here. And fairly obvious, that little bit at the top there really is just what I've kind of said on the last slide. This is just showing the amino acid turning into the zwitterion form of that amino acid. Right, properties that zwitterions have, they are highly soluble. Okay, much more soluble, and this is because of the ionic interactions. Okay, and these ionic interactions are making it soluble, are ion dipole interactions. So when I talk about ion dipole, if we look at this up here, well, that's a positive ion. Okay, water, you know, has a permanent dipole of delta negative around the oxygen, so you get ion dipole interactions there. Likewise, water, as you know, has delta positive hydrogens. Okay, and we get again ion dipole interactions down there. I think I've actually written the ion dipole interactions down here by mistake. It should be up there. Okay, they also have much higher melting points than you might anticipate. Okay, and again, that's because of the ionic interactions that you find within these zwitterions. Now, high solubility is really, really important for amino acids. They have to be transported in the blood, they have to be stored in cells. So hopefully you can see why high solubility is useful for amino acids. And that's just a little picture of some solid amino acid exists as a zwitterion, and you get these, well, it's a very small molecules, they end up as solids at room temperature. Okay, so you can pause the video and have a go at 511b, please. Okay, question 2. Question 3 again. Pause the video. Question 4. Pause the video. Question 5. Again, pause the video. Okay, so let's have a look then at the reactions of the amino acids. So the good thing about this is we've done a lot of these already, because we've done amines, we've done carboxylic acid. So these are all relevant to what we've already done. So as long as you know the reactions of amino acids, or amines, sorry, and the reactions of carboxylic acids, well, then that should be fine. So nitrous acid, if you remember, these things are aliphatic amines, okay? So because they're aliphatic amines, they do exactly the same as aliphatic amines did with nitrous acid. So they form an alcohol, the amine group turns into an alcohol group, and you get a burst of colourless, odourless nitrogen gas. Okay, so bubbles of colourless, odourless gas are your observations there. Okay, so maybe pause the video and have a go to question 3, please. Okay. That's from exercise 511D, by the way, if you're looking for it in your textbook. Please make sure you're checking all these answers. All the reactions sodium carbonate. So what would you anticipate you would see with sodium carbonate and an amino acid? Well, remember, we did sodium carbonate with carboxylic acids back in A2 module 1. So you will, of course, if it's a solid sodium carbonate, it will dissolve. You'll see fizzing of a colourless, odourless gas, which is carbon dioxide. Okay, and you could test that with lime water. Okay, have a look at this question here. So the first part of it is just pretty straightforward. The second part of it is a little bit of application. So have a little bit of a think about it. You'll have to delve back into your esterification knowledge from module A2-1. Okay, question 2, same exercise. Okay, further reactions then. So copper 2 plus ions with the amine. If you remember back even to GCSE and AS, copper 2 plus ions with excess ammonia give you this dark blue, deep blue solution. Well, the same happens with amino acids, and it's to do with, as it was with ammonia, the lone pairs really on the nitrogen of the ammonia. This uses the lone pairs on the nitrogen of the amine group. Okay, and these act as a ligand, okay, and form coordinate bonds with the 4 lone pairs of the CO negative and the NH2 of your amino acid. Now you don't probably know what a ligand is yet, so don't worry too much about what that is. We'll cover that at a later stage. Okay, you might in this instance get a bit of a blue precipitate, very hard to see in my experience, any blue precipitate. So your observation here really is the formation of a blue solution going to a deeper, darker blue solution. Okay, and that's what you get there. You can see this is 1, 2, 3, 4. Those are all dative coordinate bonds. This is the amino acid here. Okay, so we're using the lone pair of the COO negative ion and the NH2 lone pair to form dative bonds with the copper. And this complex here, which gives us this very deep, dark blue color. Okay, so pause the video, have a go at that question number 4. That is again, that's from 511D. Okay, again 511D. Pause the video. Okay, so that's as far as we'll go in video 1. Please make sure you do those questions, guys, and most importantly, as well as doing them, make sure that you check your answers, okay, before you move on.